In today's video, we're going to go over DNA technology and biotechnology. If there's one video that I honestly really wanted everyone to watch, it would be this video. This is honestly a topic that is really interesting to me and I really like, as I was thinking about doing biomedical engineering, and this just fits up right my alley. And this is a very special video because it is coming out on August 12th. And you know what that day is? National Elephant Day. So I'm gonna try. So I'm just gonna get started. It's such a good video. So first off, we're gonna talk about biotech, and there's a lot of things we can do to it. Like one thing we can do with, like, let's say DNA, we could cut it with restriction enzymes and cleave specific DNA sequences. We can also amplify it with PCRs, and it will replicate tiny DNA samples into billions of copies. You can also sequence them and determine the exact order of nucleotides in a genome. We can also edit them with CRISPRs, CAR9s, and it provides surgical precision for gene modification. That's why biotechnology or DNA technology is so interesting. So I hope if I inspire one person to pursue biomedical engineering, ah, that would be great. So when we talk about restriction enzymes, they're like molecular scissors, and these are derived from bacteria as a defense against viral phages. Phages. I don't know how to pronounce it, but these restriction endonucleases recognize specific palindromic sequences, and there's different parts to it. Like the sticky end is what's the overhanging single-stranded end that can bond with complementary DNA, and then we have the blunt end which straight cuts through both strands, and we also have DNA ligase. Ligase is like the glue, and it seals the sugar phosphate backbone after insertion. And you can see this diagram right here of restriction enzymes and like this breaking apart. You can see like the bases CTTAA and then AATTCG. Those are like the nucleotide bases. Next, we have bacterial transformation. Here's a plasmid map. Uh, First off, we can talk about cloning vectors, which are plasmids that are really small and circular, and extrasomal chromosomal DNA used to carry foreign genes into bacteria. We also have the biofactory, which once transmitted, bacteria replicate the plasmid and express the protein, such as human insulin or growth hormones. As you can see, we have the plasmid map. We see like promoter, the 5' to 3' primer site. Then we have restriction sites, inserted genes, restriction site, 3' site, original replication, antibiotic resistance, and selectable markers. So that's the plasmid map. Next, we can talk about this PCRs, the thermal cycle. The graph's a little small, so if you can zoom in, that would be great. But first, we have denaturization, where it heats up to 95 degrees Celsius, and this separates the DNA double helix into single strands. And then we have annealing, which cools to around 55 degrees, allowing primers to bond to target sequences. Finally, we have extension, where it heats back up to 72 degrees Celsius for Taq polymers to synthesize new DNA strands. And this is how, like with DNA origami, they self-assemble with each other. But I can go way in deep into that because that's just something I'm really into. But yeah. Okay, so next we can talk about gel electrophoresis. You're gonna need to know this for AP biology too. But this basically just sorts out the DNA fragments based on size and charge using an agarose matrix and electric current. So we have DNA is negatively charged, which are the phosphate groups, and migrates towards the anodes, which are positively charged, since opposite charges attract each other. and we have the shorter fragments move faster and further through the gel. So depending on the direction, the closer one will have the bigger fragments, and the one more far away has the shorter one. So that's how we separate them based on size and charge as well. And to visualize this, DNA is also stained with dyes like ethidium bromide to glow under UV light, as you can see here. Next, we can talk about DNA sequences right here. So, determining the exact nucleotide order, Sanger sequencing uses dideoxynucleotide DDNTPS to terminate chains at specific bases, which creates fragments for electrophoresis. And then the next gen sequencing, the NGS, it parallelized millions of reactions at once. And this allows a full human genome to be sequenced in a day rather than years. I hope you find this interesting. I find this interesting. Next, we have the economic shift, which is the cost of sequencing a human genome, USD. There's no way. Oh, it's in US dollars. I thought it was the acronym. But as you can see, in 2001, it used to cost 95 million. Yo, why didn't they just give that to me? But then in the modern age, this is a couple of years out of date. But right now, it's only $200 2 years ago. It's crazy. And sequencing cost has outpaced Moore's law for computing efficiency. Yeah, I'm not gonna be a finance major. That's too difficult for me. Okay, so now we're gonna talk about CRISPR Cas9 editing. So this is used with like, no, this is a surgical edge, and it's a specialized system that's adapted for bacterial immune mechanisms to provide targeted gene editing. So we have the Cas9 proteins, which are the endonucleases that perform the cut, and then we have the guide RNA, the gRNA, which directs this Cas9 to a specific 20 nucleotide sequence in the genome, and then repair. The cell's natural repair mechanisms fix the break, allowing the scientists to disable genes into new ones. You can see, it's a little small here too, but if you would like to pause and like zoom in, you should do that so you can understand what's happening here. Next, we have real world examples with biotech in medicine. So let's say we have recombinant insulin, and this is a bacterial expression of a human gene, and it's a constant supply for diabetics. Like some people can't produce enough insulin, or some people don't have a reaction to insulin, and that's basically what diabetes is. We also have mRNA vaccines, which are direct injection of genetic instructions, and this is a rapid pandemic response for COVID-19. So this is all biotech, biomedical stuff. And then we have gene therapy, which is a viral vectors that delivers functional genes. And this is a potential cure for genetic disorders. I'm sorry. And then we have diagnostics, which are qPCRs, which are fluorescence-based DNA detection. And this goes on with like biosensing that has to do with DNA technology. And this is like early detection of viral/bacterial loads. Super interesting, as you can see, like really life-changing stuff. We also have a stem cell. So they have a regenerative potential, which means undifferentiated cells are capable of renewing themselves after differentiation into specialized cell types. We have like tetipotent, which can become any cell type plus central tissue. We also have pluripent, which can become any body cell type. And finally, we have induced pluripotent, iPSCs, which adult cells reprogram back to a stem state, bypassing ethical embryonic concerns. There's another graph right here representing what I just said. Make sure to pause to look at it. We also have GMOs in agriculture. I'm not sure what Jerry was trying to do, but GMOs is just genetically modified stuff. Also, we have DNA fingerprinting, as you can see here. The number of genetic location analyzed to create a unique profile. So this is like how people the like police match suspects with like DNA from a crime scene. So the first step, this is good for AP biology too, but you go here and then you look to the right to see which one matches the most with all of these, and then which one matches the closest. It might not match completely, but whichever one like matches the most is probably the likely suspect. And as you can see here, suspect 2 has basically the same locations, and this would match him with the crime scene, which would help the police catch him and prove that he was guilty for the crime. With every field, there's ethical frontiers, and there's a quote that goes along with this. We must not only decide what we can do with biotechnology, but what we should do. The power to edit the human germline is a power over the future of the species. And that just basically says that this is the potential for the future, but we have to understand like the cost that could be associated with it and understand what we're doing, as this has the power to change the human germline, and the power is within us to make sure we get those decisions right. So, any questions? Leave in the comment section. I know a good amount about biotechnology, DNA technology. I'm gonna definitely go in more depth about specific nanotechnology, but this is like a really basic overview. If you have any questions, leave them in the comments, and I'll try answer them to the best of my ability. And we have sources right here. And yeah, I hope you have a great day, and I'll see you later. Bye.